Welcome to Vet Neurojar, the podcast dedicated to exploring the fascinating world of veterinary neurology and pathology. Today we're diving into a very rare and intriguing case, Anaplastic Oligodendroglioma in a rabbit. This is a first, so buckle up. Recently, a group of veterinary pathologists from Osaka Metropolitan University in Japan documented a unique case that has broadened our understanding of neurological diseases in domestic animals. Their work, detailed in the Journal of Veterinary Medical Science, reports the occurrence of anaplastic oligodendroglioma in a 6-year-old male rabbit. Now, before we get into the specifics, let's set the stage. Brain tumors are uncommon in pet rabbits. In fact, the scientists noted that only one prior case of a brain tumor, a glioblastoma, has been reported in rabbits. But oligodendroglioma? This is the first instance of its kind. Our patient, the 6-year-old rabbit, presented with some alarming symptoms. He had lost his appetite, was rotating to the right side, and circling to the left. Initial suspicion fell on encephalodozoonosis, a common parasitic disease in rabbits, and treatment was started accordingly. Unfortunately, the rabbit's condition didn't improve. Renal failure was detected the next day, and despite supportive care, he died just 2 days after the initial presentation. This rapid decline underscores the aggressive nature of the underlying condition. A necropsy, or animal autopsy, was performed to determine the cause of death. A red to orange lesion was found in the right bottom of the midbrain. Further examination revealed a poorly defined, whitish-gray lesion, about 1.5 centimeters in diameter, containing areas of hemorrhage, extending from the cerebrum to the midbrain. Samples were collected from various organs, including the brain, and prepared for detailed microscopic analysis. This is our first checkpoint of the show. Question 1. What differentials would you consider based on the symptoms the rabbit showed at presentation? Keep that in mind as we continue.
Histological examination, or the microscopic study of tissue, revealed a high cellularity area within midbrain. This means there was a dense concentration of cells in the affected region. Neoplastic cells, or tumor cells, were seen proliferating, displaying large, irregular nuclei and varying amounts of cytoplasm, a hallmark of anaplastic tumors. The cells grew diffusely in sheets, with some areas showing a sudocet arrangement, also known as homerite rosettes. The cells exhibited moderate atypia, meaning they deviated significantly from normal cells, and mitotic figures, or cells undergoing division, were frequently observed. Key 67, a protein associated with cell proliferation, was present in more than 10% of the neoplastic cells, confirming the tumor's rapid growth rate. The tumor was invasive, extending into surrounding tissues like the thalamus and hypothalamus. Hemorrhage and angiogenesis, or the formation of new blood vessels, were common within the tumor. Necrosis, or tissue death, was sparse. Interestingly, microgranulomas, small clusters of immune cells associated with encephalozoonosis, were also present in the cerebral parenchyma, adding complexity to the case. To further characterize these neoplastic cells, the team performed immunohistochemistry, a technique that uses antibodies to detect specific proteins in tissue sections. The results were quite revealing. More than 80% of the neoplastic cells showed nuclear labeling for Olig-2. Now, Olig-2 is a transcription factor crucial for the differentiation of neural precursor cells into oligodendrocytes. At the same time, the neoplastic cells tested negative for myelin basic protein, or MBP, and PLP, markers of myelin-associated proteins. Less than 10% were positive for SOX10, an oligodendrocyte precursor cell marker.
The scientists also looked at cytoskeletal proteins, performing immunohistochemistry for nestin, vimentin, and GFAP. More than 80% of the neoplastic cells showed cytoplasmic labeling for vimentin. These vimentin-positive cells were also positive for Olig-2. Only a few GFAP-positive cells, resembling reactive astrocytes, were present in the highly cellular tumor area, while the neoplastic cells themselves were GFAP-negative. This brings up our second checkpoint. Question 2. How do the histopathological and immunohistochemical findings contribute to the classification of this tumor as anaplastic oligodendroglioma rather than another type of glioma? Really put your thinking caps on, folks.
Finally, transmission electron microscopy, or TEM, was employed to analyze the ultrastructure of the neoplastic cells. The cells had round, electron-dense nuclei and a moderate amount of cytoplasm, resembling oligodendrocytes. The cytoplasm contained randomly arranged short fibers, measuring 10 to 12 nanometers in diameter, consistent with intermediate filaments. This finding supported the immunopositivity for vimentin. Now let's pause and consider what all of this means. The tumor's characteristics, including high cellularity, nuclear pleomorphism, frequent mitotic figures, and positivity for Olig-2 and vimentin, align with the criteria for Anaplastic Oligodendroglioma. The ultrastructural features further supported the classification, with the cells resembling immature oligodendrocytes. Anaplastic Oligodendroglioma, also known as high-grade glioma, is a malignant tumor that exhibits aggressive growth and invasion. In domestic animals, the classification of central nervous system tumors often follows the World Health Organization's WHO classification system used for human tumors. In dogs, anaplastic oligodendrogliomas are characterized by the proliferation of immature and anaplastic oligodendrocytes, displaying epithelioid morphology, large and pleomorphic nuclei, and abundant cytoplasm. High cellularity, multifocal hemorrhage, necrosis, and angiogenesis are commonly observed. Canine anaplastic oligodendrogliomas typically express oligodendrocyte precursor cell markers like Olig-2, SOX10, and PGFR-alpha. This suggests that these tumors originate from OPCs.
The neoplastic cells in the rabbit case shared similarities with immature oligodendrocytes, exhibiting invasive growth and malignant behavior. The cells were immunopositive for both Olig-2 and vimentin. Olig-2 plays a crucial role in differentiating neural precursor cells into OPCs. In the central nervous system, neural stem cells and glial precursor cells express nestin and vimentin during differentiation. As these cells specialize into oligodendrocytes, they typically lose these intermediate filaments. The characteristics of the neoplastic cells in this case align with those of immature oligodendrocytes, resembling OPCs. Although PDGFR-alpha is a well-characterized marker for OPCs, the antibody did not cross-react with rabbit tissue, complicating the analysis. Myelin-associated proteins like MBP and PLP are not consistently detected in canine oligodendrogliomas, which is consistent with the immunohistochemical results in this case. Glioblastoma, another aggressive brain tumor, was considered in the differential diagnosis. While Olig-2 is often associated with oligodendrogliomas, it is not always specific, as canine astrocytomas can also express Olig-2. Additionally, vimentin is sometimes associated with astrocytomas in humans. In this particular case, the neoplastic cells showed a high proportion of Olig-2, immunopositive and GFAP immunonegative cells. Based on these ultrastructural and immunohistochemical features, the diagnosis of anaplastic oligodendroglioma was confirmed. Let's take another pause to keep us going strong. Question 3. If you could perform additional tests that could further refine your understanding of the tumor, what would they be and why? Write down your answer and share them with your fellow neurology and pathology lovers.
The authors of the study acknowledge that this is the first reported case of anaplastic oligodendroglioma in a pet rabbit. They emphasize the need for further studies to determine if the histological features observed in this case are typical of oligodendrogliomas in rabbits. This case underscores the importance of considering rare and unusual conditions in veterinary medicine. It also highlights the value of detailed pathological investigations in advancing our understanding of disease processes in animals. By sharing these findings, veterinary professionals can be more vigilant in recognizing and diagnosing similar cases in the future. Finally, this brings us to our last engaging question. Question 4. What are the implications of this case report for future research on brain tumors in rabbits and other small animals? Well, that is all the time we have for today. Thank you so much for joining me on this journey into this rare case. The original article is available in the Journal of Veterinary Medical Science, in case you want to dive into the research. From Vet NeuroJar, keep those minds inspired, hearts light, and tails wagging.